Hello guys, welcome to BIOGURU. Now we will discuss about the DNA fingerprinting. So what is DNA fingerprinting? A technique used by the scientists to distinguish between individuals of the same species using DNA as a sample. And it was first introduced by Alice Jeffrey in 1984. So DNA fingerprinting, also known as DNA typing or genetic fingerprinting, is a method for identifying individuals by the particular structure of their DNA, because we have a unique sequence of DNA. DNA fingerprinting is used for many purposes, particularly paternity, maternity testing, and for forensic work. So, DNA can be obtained from whole blood, semen, vaginal fluid, hair roots, almost any tissue, and even from bones that have been buried for a long time for fingerprinting. So, now there are many samples and procedures. So, DNA fingerprinting is a technique by which the DNA of an individual can be compared with that found in a sample और another individual, a suspect in a crime. तो एक इंडिविजुअल का डीएनए दूसरे इंडिविजुअल से मैच करते हैं. कभी भी दो इंडिविजुअल्स का डीएनए आपस में मैच नहीं कर सकता जब तक कि वो आइडेंटिकल ट्विन्स ना हो. सो ATGC are dash of DNA and two bases, sugars, ions or none. It is N2 base. That's great. So now DNA fingerprinting protocol and advantages. So first of all, DNA fingerprinting, the technique is carried out as follows. DNA obtained from blood, semen, hair root sample, etc. So isolation of the sample. Then isolation of DNA. If DNA obtained is very less in quantity, then it is amplified using PCR. PCR is polymerase chain reaction. Then DNA fragmentation. Now DNA के छोटे छोटे fragments करेंगे. DNA sample is fragmented using restriction endonucleases. Then many fragments are obtained having variable length. अलग अलग length के बहुत सारे आपको क्या मिलेंगे? DNA fragments और segments. Restriction fragment length polymorphism, RFLP. So नाउ PCR के थ्रू हम डीएनए को क्या करेंगे? PCR के थ्रू आप डीएनए की नंबर ऑफ कॉपीज जनरेट करेंगे. देखिए देयर आर सम प्रोसेसेस. फर्स्ट इज डिनेचुरेशन. डिनेचुरेशन से डीएनए को सेपरेट किया गया 94 डिग्री सेंटीग्रेड पर. देन अनीलिंग. अनीलिंग से प्राइमर्स को उसमें ऐड किया गया. एंड देन एक्सटेंशन. एक्सटेंशन से उन प्राइमर्स को या कहा जाए डीएनए प्रॉप्स, उनको क्या किया गया? एक्सटेंड किया गया. So there are three processes जैसे हमने पढ़ा: denaturation, then annealing and extension. Denaturating DNA, the DNA on the gel is denatured using alkaline chemicals or by heating. तो DNA को आपने separate किया. Then denaturing means separation, splitting of double helix into single strand by breaking hydrogen bonds between the two strands. Then the fragments are then separated by agarose gel electrophoresis. Then gel electrophoresis. So it is agarose gel. Then there are some wells. Solution. So अब इनको आपने क्या किया कि separate किया according to the charge. So DNA samples placed in well cut in gel. तो जो जेल में कट करके आपने छोटे छोटे वेल्स बनाए थे, वहां आपने उन DNA samples को फिल कर दिया. Then voltage लगाया गया एनोड, बिकॉज DNA have negative charge. So हमने यहाँ पे एनोड लगाया एंड नेगेटिव इलेक्ट्रोड एंड पॉजिटिव इलेक्ट्रोड. अब DNA मूव करेगा टुवर्ड्स द पॉजिटिव इलेक्ट्रोड. द मूवमेंट ऑफ दीज फ्रेगमेंट्स डिपेंड ऑन देयर लेंथ. देखिए नाउ वेल थे, अब ये मूव करना स्टार्ट करेंगे. 20 मिनट्स, 30 मिनट्स, 50 मिनट्स, 60 मिनट्स, 70 मिनट्स, DNA का मूवमेंट होता जाएगा. जितने भी लार्ज फ्रेगमेंट्स हैं वो पीछे की तरफ आएंगे, और जितने भी छोटे फ्रेगमेंट्स हैं, शॉर्ट फ्रेगमेंट एंड लाइट फ्रेगमेंट्स होंगे, वो आगे तक मूव करेंगे. सो दिस रिजल्ट्स इनटू फॉर्मेशन ऑफ बैंड्स. तो वहां डीएनए के बहुत सारे बैंड्स बन जाएंगे. द बैंड्स आर देन ब्लॉटेड ऑन नाइलॉन मेम्ब्रेन, नाइट्रोसेलुलोज मेम्ब्रेन. उनको हम ट्रांसफर करेंगे. देन सडन ब्लॉटिंग आपने क्या किया? ट्रांसफर किया उसको नाइलॉन मेम्ब्रेन पर. and single-stranded DNA gets embedded into nylon membrane. And as the towels draw moisture, the DNA strands are transferred onto the nylon membrane by capillary action. This process is called blotting, more precisely Southern blotting, after the name of its inventor, E.M. Southern. So, hybridization कराया गया. The bands are flooded with single-stranded radioactive probes. जो radioactive probes बनाए गए थे, उनसे हम उन्हें मैच करेंगे. Probes get complemented to the complementary VNTR sequences. तो जो probes हैं, वो अपने complementary sequences के साथ complement कर जाएंगे. The probe attaches by base pairing to those restriction fragments that are complementary to its sequence. The probes can be prepared by using either fluorescent substances or radioactive isotopes. So, एक्सेस प्रोब्स आर वॉश्ड आउट. जो एक्सेस प्रोब्स थे उनको आपने वॉश आउट कर दिया, रिमूव कर दिया. देन फोटोग्राफी फिल्म लेंगे इसकी. द नाइलॉन मेम्ब्रेन इज केप्ट इन कांटेक्ट विद एक्स-रे फिल्म. देन डीएनए बैंड्स ड्यू टू रेडियोएक्टिव प्रोब गिव फोटोग्राफिक इमेज ऑन एक्स-रे फिल्म. And the thick and thin dark bands form a pattern of bars which constitute a genetic fingerprint. Now this is how the developed X-rays film looks like. You can see in the images. Autoradiogram. Now, parental disputes, catching criminals and forensic science में हम DNA fingerprinting को use करते हैं. Now, isolate chromosomal DNA from a sample of cells. Then sample 1, sample 2. Sample 1 cells obtained from blood, hair or semen. Sample 2 cells obtained from blood, hair, roots or semen. Then second step, cut the DNA with the restriction enzyme. Separate the DNA fragments by gel electrophoresis. Then agarogel electrophoresis, decreasing size. Now, transfer bands to nitrocellulose. Then stack of paper towels, nitrocellulose, filter paper, salt, solution gel आपने use किया. Then nitrocellulose with transfer DNA bands, liquid containing radioactive probe, cDNA, RNA or cloned genomic DNA. Denature the DNA and add probe, allow probe hybridize and wash away excess probe. Radioactive probe hybridizes to nitrocellulose filter. Then hybridized nitrocellulose washed out and autoradiographed. Then expose the filter, the photographic film and compare the result. Exposed film, this one. So, a given person can never have a VNTR which his parents do not have. तो किसी भी पर्सन में अगर पेरेंट्स में वो VNTRs नहीं है तो वो कभी भी नेक्स्ट जनरेशन में ट्रांसफर नहीं होते. Obtaining hybrid with radioactive probe and matching DNAs of different members of family with biological children and adopted children gives us an idea of how DNA fingerprints help identification of paternity-maternity by studying the DNA fingerprints of members of a family, biological and non-biological relationships. The illustration given below are the VNTR pattern for Mrs. Rose, blue, Mr. Brown, yellow, then D1, Mr. Rose biological daughter, then D2, child of Mr. Rose and her former husband, and S1, Mr. Rao's biological son, and S2, Mr. Rao's adopted son. Not biologically related, his parents' DNA marked in light and dark green bands. ये हम एक छोटा सा यहाँ पे एग्जाम्पल ले रहे हैं. So mom, dad, D1, D2, S1, S2. D1 is daughter 1, D2 is daughter 2, S1 son 1 and S2 son 2. So selection of victim DNA. Then 24 repeats of GT, 16 repeats of 80 GG and 66 repeats of 8 TT. There are 3 regions of repetitive DNA. Section of suspect's DNA. Then यहाँ पर हमने लिया again 49 repeats of GT, then 16 repeats of ATGG and 51 repeats of ATT. The same three regions of repetitive DNA are present here, but some include different numbers of repeats. Now let's compare the sample too. Section of DNA from crime scene here. Then next 49 repeats of GT, 16 repeats of ATGG and 51 repeats of ATT. The length of repetitive sequences match the lengths in the suspect's DNA. So the DNA found at the crime scene belongs to the suspect. So now applications of DNA fingerprinting. Conservation of wildlife, protection of endangered species by maintaining their DNA record for identification of tissue of the dead endangered organisms. Then taxonomical applications, study of phylogeny for evolutionary knowledge. Pedigree analysis, inheritance pattern of gene through generations. Anthropological studies, charting of origin and migration of human population. Then medico-legal cases, establishing paternity and or maternity more accurately. Forensic analysis, positive identification of a suspect in a crime. Genome is now some MCQs. Genome is a technique for DNA sequencing, complete set of gene present in an organism, particular segment of DNA or mutated segment of DNA. Yes, it is complete set of genes present in an organism. Human Genome Project was initiated by US Department of Agriculture, Energy, Science and Technology or Health. It is energy.